So just to remind you of things that you need to know for this model, the whole thing here is the neuromuscular junction. This part here is the motor neuron. This whole thing here is the motor neuron. All of this is just a section of muscle fiber. Okay, this is just a section of a muscle fiber here. When I make the decision to move a muscle, that action potential is going to be brought down the axon. So here in yellow, I've got the axon of the motor neuron. In yellow here, I also have the axon of the motor neuron. That action potential is going to go down towards this area. Once I start seeing these little dots here, at this point, we are at the axon terminal of the motor neuron. Okay. So that depolarization in the form of sodium ions comes down here. It's going to depolarize the axon terminal. Voltage-gated calcium channels open up. Calcium comes in. Calcium unbinds these vesicles from their docking sites, and then they're able to fuse to this here. This white thing is the presynaptic membrane. Okay, so these vesicles are going to fuse to the presynaptic membrane and dump acetylcholine into the synaptic cleft. The synaptic cleft is the space between the presynaptic membrane and the postsynaptic membrane. In this model, the postsynaptic membrane is this convolution here that's tan, so that would be the postsynaptic membrane. And this is the postsynaptic membrane of the muscle fiber itself. Okay. Now remember, it's the postsynaptic membrane that has the nicotinic acetylcholine receptors. They actually don't show the receptors on here, but they do exist. When the acetylcholine binds to these receptors, the receptors open their sodium channel, and I get a localized depolarization of it, right? After that, if I get enough depolarization, then my voltage-gated sodium channels are going to open and action potential occurs. When that action potential occurs, it's going to spread throughout the sarcolemma. Here's part of the sarcolemma here. Okay. The postsynaptic membrane is going to be next to the sarcolemma. Okay, this is also oops, let me do that better. This is also sarcolemma here. Okay, so it goes down throughout the sarcolemma, and the action potential then goes deep into the muscle fiber through these blue things here. These are Ttubules. Okay, they go down the Ttubules. And these here are truncated. They're just cut off Ttubules. All of the red things here are mitochondria. They're all mitochondria. Okay. Once the action potential gets to these Ttubules, here in tan, I've got the sarcoplasmic reticulum. The sarcoplasmic reticulum is going to then open its voltage-gated calcium channels, and calcium is going to leave the sarcoplasmic reticulum, which allows, if you recall, which allows the cross-bridge cycle then to occur. A couple other things that you need to know. So recall that this whole thing here is just a section of the muscle fiber. So each one of these is a myofibril. This whole thing is a myofibril. This whole thing is a myofibril. And you might recall myofibrils have actin and myosin. Here I've got a Z line. So here's a Z line, here's a Z line from here to here. That would be a sarcomere. A sarcomere. Okay. A couple other things to point out. This is a nucleus of the muscle fiber. Nucleus of the muscle fiber. This is the nucleus of a Schwann cell. Okay, now this that wraps around the axon of the motor neuron, this is called the myelin sheath. The myelin sheath. Okay. The myelin sheath is created by the Schwann cells. To get a better look at some of these Schwann cells, here in the back, This is a Schwann cell. This is part of a Schwann cell here. All right, so Schwann cells have these big arms that wrap around the axon. So here is a nucleus of a Schwann cell, and here is the nucleus of a Schwann cell. That also is depicted by this blue thing here. If I turn this around, so you might recall that this is the sarcolemma, right? The sarcolemma here. This part is actually the endomysium, and it's made up of two layers. Okay, it has the basal lamina, and it has reticular fibers. So these are reticular fibers.